It's a great pleasure to be here. I always enjoy coming back to Seattle. I started my career here many years ago at the University of Washington, and it feels like I'm coming home every time I come. And I've never been to the Institute before, and so I really enjoyed visiting here. I'm going to be talking about something called antimaps, and this talk is going to have, I'm done at 10 of, right? Yeah. This talk is going to try to answer 5 questions. What is an antimap? Can it exist? I'll tell you, when I tell you what it is, you'll wonder maybe it couldn't exist. How would you make an antimap? Could the brain have antimaps in them? And why in the world would you want one? Before I talk to you about antimaps, let me tell you a little bit about maps in the brain. You can see that, okay? So the best understood part of the brain is probably the visual cortex, and here is you're looking at the back of a monkey brain. The reason it's a monkey brain is because if you looked at the back of our brain, you wouldn't see much visual cortex. It's all buried. But the monkey brain is much less wrinkled. And so you can, when you look at the back, you see the whole visual cortex right there, primary visual cortex. Here is visual field of the animal. This part of the visual world goes to this part of half of the visual cortex. This half goes there, and you can see, I can't see it, but I presume you can see that there are a lot of concentric rings here, and those concentric rings are the lines that go vertically over there, and the lines that are going radially are the ones that go like this. The center of vision is over here, and the periphery is up that way. So if I record from a cell at a particular location here, I know where that cell is looking in the world. Not only that, but if I record from a cell here, if I take a little piece of the cortex here and I expand it, it has a structure that are called pinwheels, and the pinwheels, here's one here, a pinwheel. And this is a little more regular than the real cortex is, but if you look at a real cortex, you can see these pinwheels on their surface. And as you go around the pinwheel in this way, you encounter cells that have a horizontal, and then they'll gradually go to 45 degrees and then vertical. So cells respond to a particular orientation of an edge. And as you go around the pinwheel center, what you get is cells that respond to various corresponding angles of an edge. And as you go from the pinwheel center out one of these spokes, this would be the horizontal ones here at 0 degrees. As you go out that way, you have cells that have very fine and then much bigger receptive fields, so that they are looking at very fine features and very large features. The point of this is that if you know where you are in the map, if, for example, I'm right here in the map, then what I know where in the map tells you what information that cell conveys. This is a version of location is destiny. An antimap is a region of the cortex, say, a region of the brain, where it is the opposite in a certain sense of a map, opposite in the sense that telling you that knowing where a cell is gives you absolutely no information about what that cell is doing. So if I draw here a region of the brain, and this region of the brain is some particular part, and I'm going to give you an example later where there may be an antimap. I'm going to claim there is, and that antimap will be in the anterior piriform cortex. But other places where I believe there are antimaps, as you'll hear later, there is an anatomical signature for antimaps, and other places that have that same anatomical signature are the hippocampus and in the cerebellum. So if I record from a critical number of cells, N, and I record from that cell, that cell, that cell, that cell, and that cell, in this particular case, the critical number N is, in that cell, is 6. I get all the information that's available. But if I record from this cell, this cell, this cell, this cell, this cell, and that cell, I get just the same information. And if I record from half of the green and half of the white, the same information. So the thing about an anti-map is sort of like an anti-hero in a novel. Telling location is not destiny at all. Destiny is how many cells you record from. You record from a certain number, you know it's there. Okay. That's what an anti-map is. The question is, can it exist? Is it possible to make an anti-map? Might not be. It seems a little funny that you could record from any cells and get the same information. How would you do such a thing? Okay, now I'm going to take a step aside and tell you about a new field of mathematics and computer science called compressed sensing, or sometimes called compressive sensing. This field is around a decade old. The first papers were published about a decade ago, and it has taken the world of computer science and certain areas of mathematics by storm. So many people in many departments of computer science work on various aspects of this. And to humanize it, I'm going to tell you about the main person, Terry Tao. He's in the mathematics department at UCLA. I'm going to tell you about Terry Tao. And I recommend to you, if you want to waste some time, go to Wikipedia's biography of Terry Tao and read it. It's really fun to read. Terry Tao was a first-generation Australian of Chinese parents, and he came to the United States to Princeton for his PhD. He got his PhD when he was 20 years old and went to UCLA. When he was 24 years old, he became UCLA's youngest professor ever, full professor. And I think I can't remember exactly, I haven't looked back his article at his biography, but I think he got the Fields Medal, which is the sort of Nobel Prize of mathematics, when he was 28. And he's incredibly productive, and compressed sensing is just a small part of what he's done. Here's the problem that Terry Tao started. Up here you have an image, for example. And if you take an image picture with your camera, and then you save it on your computer or save it in the computer memory on an SD card, it's compressed. There's a particular way that you compress it. And so you start with an image. And then you make a compressed version of that image. And I'm not going to say much in detail about it. Basically, the way you do it is you do a particular mathematical, for MPEG images, you do a particular mathematical operation, which is it's called a wavelet transform. It doesn't matter, but it represents the image with a whole lot of coefficients. You then, not you, the camera, computer, looks over that image. It finds all the small coefficients, throws them away, sets them to 0, keeps the big ones. And usually, for most images, it depends on what the image is, but in general, any image you take, you can compress it if you keep like somewhere between 2.5% and 5% of the coefficients. Then you do, this is sort of the dehydration step. And then you can do a rehydration step. And the rehydration step is you do what's called, you take all the zero numbers, you put them in zeros instead of the small numbers, coefficients that used to be there, and you do what's called an inverse wavelet transform, and you get your image back. So Terry Tao said, you know, this is a lot of work. You have to take in all the, 8 megapixels worth of data from your camera, and then you have to look at all 8 megapixels, do a big operation on them, which is the wavelet transform, look at all those coefficients. He said, wouldn't it be nice if we had a way where we could just not have to look at all the pixels, but have an automatic way of taking any image and dehydrating it. So you don't have to think about it. You just do it automatically. And in fact, if you were clever enough, what you could do is you could take in just the data that you needed, and you wouldn't even have to take all 8 megapixels in. You could compress it on the fly. And he figured out, and what he proved is that you can dehydrate an image in a particular way, and I'm going to tell you what that is in the next slide, and rehydrate it and almost surely get it back almost perfectly, but perfectly if you don't dehydrate it too much. This is the process of compressed sensing, and it's starting to be used now. So, for example, there is a way of taking in data from MRIs, and so that you can take in about 1/6 of the data. So that means you do your MRI 6 times faster, and you get the same resolution that you would have had you taken the whole thing in and then compressed it later. Okay, how does this work? The image is a bunch of pixels. In a expensive camera, it's 8 megapixels. And what you do is you take all those pixels and you just string them out in a long line of numbers. And here is, and that's called the signal. And here is a long line of numbers. Actually, I didn't want to quite do that, but. And here's the first one, and there's the second one, and there's the third one, and this is the nth one down here, and you have n numbers. Then you make a big array of numbers, and I'm going to tell you how you do that. And this array is n cells in this direction. Here's the first, here's the second, here's the nth. And it is m cells in this direction, first, second, and mth. And so this is an n by m array. It's a matrix. And what you do is you fill all of these cells in the matrix with random numbers. Gaussian distributed zero mean random numbers. But they don't have to be that. That's what you do. And then you multiply, this is a matrix, you multiply this matrix by this long list of values over here, that's a vector. You multiply it, and what you do is you take the first entry here, you multiply it by the first entry in the signal. Take the second entry here, you multiply it by the second entry in the signal. You do that one by one all the way through, the nth one here times the nth one there, and then you have n products, and you add them all up. And then you write them down over here in the output signal, in the output vector. And the output vector here is, so what you did is you take the first row here, multiply this times this, this times this, this times that, add them all up and write it down there. Then you go to the second row here, And you do the same thing again, first times first, second times second, up through nth times nth, and you write it down in the second one here. And that is matrix multiplication. And so this is your compressed signal. So what Terry Tao proved is that if you make, this is called a random projection. What Terry Tao proved is that if you take random numbers here, and if you do this multiplication and put it over there, if m is less than n, but it's big enough, and big enough is difficult to define, but by experience you can figure out, it's sort of like doing the compression with your camera. There are certain numbers that work really well, and you can make it better if you need to. Then he proved that you can take this signal over here, which is maybe shorter by m, and get your signal back perfectly. So you lose no information in this process, and you compress it, or essentially no information. Okay, so what this means is that if m is less than n, then, but if m is big enough, then you have compression. And you don't have to ever look at what you're doing, and if you're clever, you don't even have to take in all the data, you can get it. So this is compression on the fly. This is called compressed sensing because you're sensing the environment and compressing it as you sense it. Or maybe take it all in, but not have to look at it, just compress it automatically. If m equals n, this is compressed, CS, compressed sensing here. If n equals m, all you've done is taken your data and multiplied it by a lot of random numbers. But what you can prove, and extremely easy, I'm not going to take time, then you can get back your signal perfectly, almost surely. This is perfect sensing. And if m is greater than n, in other words, you don't compress, but you expand, you would call this expansive sensing, expanded sensing. And that has an interesting property. Suppose that if going down to here is enough to get your signal back. In other words, this m is long, but you only need the first 40 of these, say, to get your signal back. Then, if you look at the first 40, well, you get your signal back. But these are all random numbers. It's always the same signal. You look at any 40 here, you can get your signal back. And you can look at the first half of these and the next half down there, get your signal back. This is, I've done two things. Terry Tao did it, proved that you can have an anti-map because it's just, all you do is you take your signal, you multiply it by a big random matrix, and that gives you an anti-map because you can get the signal back from any certain number over here. And it tells you how you could do it. Now, the point is, that I'm about to make, is that the olfactory system naturally does this. Its anatomy just has this built in. And this is a caricature of an olfactory system with two odorant receptor genes. There's a white gene and a green gene. This is the nasal epithelium, sensory epithelium, and here are four white cells, all having white receptor, and here are four cells having the green receptor. This were a mouse. Those there would be 5 million of those that we heard this morning, and they would have about 1,000 different genes. All the green ones go over to the bulb, and their axon terminals all are in a place called the glomerulus. They're all lumped together. And all the white ones go to another glomerulus. So this bulb has two glomeruli. In a mouse, there'd be 2,000. each bulb has multiple mitral cells. In the mouse, around 50 mitral cells go into each glomerulus, but in this poor animal, there are only 2 mitral cells here and 2 mitral cells there. Their axons go over to the cortex, and this cortex has 1, 2, 3, 4 input neurons. And over here, they make synapses at random. One there, none there, none there. Here's there. This one has 3. And so what happens is, as this axon goes along, it comes to each mitral cell. The mitral cell axon goes along. It comes to each layer 2 neuron in the olfactory cortex. It flips a coin. It says, shall I make a synapse or not? If it gets heads, it makes a synapse. If it doesn't, here it got heads, no heads. There it got heads. Here it got no heads, heads, heads. Here it got, this green one got heads here, not there, heads there, not there. It's a coin flipping problem. How many synapses you make, it's just coin flipping. That's random. And so what's happening is that the strength of the signal from this glomerulus onto the cortex is determined by random, by how many coin flips come up. And so, for example, there are 2 here, there's 1 there, there's 1 there, and there's 0 there. So let me write it down. We're actually almost done. Okay, so here is the bulb. And there are 2 glomeruli, the white and the green. And here is a synaptic strength matrix, which is 2 this way and 4 this way. And over here is an expanded vector, which are the cells in the olfactory cortex. This is white. This is a random number here, 1. This is a random number there, 2. Well, let's see what they are. Okay, so the first row here, there's going to be 2 entries, 1 for this, 1 for that, and you multiply this times that and this times that, and so the number of synapses that are made by the white thing here, in this case, is 1, and the number of green ones in this number of white ones for here is 1. The number of green ones, the total number for green ones is 1. So we have a 1 and a 1 there, and then let's go to the third one over here. We would have 2. Here would be 2 there and 1 there, and the second one would be 1 and 0. So so the weight that's assigned to this glomerulus, the white glomerulus, is 1, and so this gets 1 synapse. The weight assigned to this one is 1, and so this is weighted 1, this is weighted 1, but the currents from both of those synapses get added together. So it's 1 times this plus 1 times that equals the first thing over here. That's what goes into the first cell. Then you go to the third one. There are 2 here. This is the third cell down. There are 2 in the cortex. The weight is 2, so you multiply this times 2, that times 1, and then the cell adds up the current. It adds it up for you. And so the third one, so the first entry up here would be 2 times whatever is in the bulb. The next one would be 1 times whatever in the bulb. The next one would be 3 times whatever is coming from the bulb. And so what I just did is I just showed you that what the synaptic weight matrix does, it automatically takes whatever signal is in the bulb, it weights it by random numbers over here, adds those all up and puts them in the cells over there. That is exactly what the expanded sensing does. And so, just in its anatomy, the olfactory system has to be doing expanded sensing, which means that it forms an antimap, which means that you can look at any of the cells in the cortex. If you look at two of them, you'll get all the information that's possible. Okay, why would you do this? There's not time. You can ask me later, but basically it has to do with this. Evolution can plan in things for the visual system because there are many constraints in the environment that it could make uses of. But if you have odors, there's no way of telling which odors are going to be matched up. And there's no way that evolution can plan that in. So basically you have a high dimensional space of odors. The brain has to be able to put a label on any point in that high dimensional space. And in order to do that, it has to have lots of substrates for doing it with. And by doing expanded sensing, all the information is there, but you generate lots of substrate for this. An example of this is coffee. Nobody drank coffee until 500 years ago. Starting 300 years ago, everybody was drinking coffee. Everybody knows the odor of coffee. There's survival value in it. A lot of people say that if you don't have your coffee, you're not going to make it through the day. And yet, evolution could not possibly have planned in that we need to know the odor of coffee because it wasn't even there over most of human evolution. And so that's a trivial example, but there are many examples where you can't plan, evolution can't plan, so the brain has to be able to learn it, and this is the ideal system for making a substrate for that. Okay, thank you. I'm sorry I went over.